Yeah, so good evening to everyone who has already joined us. Today we have the fifth meeting of our session of seminar series of seminars in which we discuss genetic variation analysis. We've already covered basics of variant calling in germline and somatic settings. We've talked about copy number structural variants. We've last time had the discussion of genome-wide association studies and issues related to these types of analysis. And today we continue our journey, and now we'll be talking about population history reconstruction. So initially we were planning that this meeting will be more about ancient DNA analysis, but in fact we would like to cover a broader range of topics and to focus a little bit more on various statistics for population history, and we'll also cover briefly some of the aspects which are related to paleogenomic data analysis. Yeah, and as the main speaker today, I'm pleased to introduce Dr. Pavel Flegontov, who will, I hope, say a few words about himself. Yeah, Pavel, please.
Okay. So I've been working in archaeogenetics for more than 10 years. Now I'm a group leader at the University of Ostrava in Czechia and in the Czech Republic, and I'm also affiliated with the Reich Lab at Harvard University. And I started working originally in just reconstructions of population history, but now I'm moving into the area of method testing mostly. So I don't study history of various populations. I just study most popular methods used to study that history.
Okay, thank you. Yeah, and I think that we can jump right in with the main part of the seminar. So yeah, I think that I can right away give floor to you for the introduction to the topic, and yeah, we'll follow up with my questions and the discussion as we go along.
Yeah, so sure. I decided to structure my presentation into two parts. First part will be based on our recent published results. And the second part will be more speculative. It will be based on very fresh, unpublished results, but I think they're interesting for people working in PopGen because we will discuss PCA, a method that everybody uses. Okay, so do you see my screen?
Yeah, we do.
Okay, good. Okay, so this title actually refers to the second part of the talk. So let's start from the beginning. There are many potential sources of bias in archaeogenetic research because there are many layers of archaeogenetic research, starting from ancient DNA isolation and sequencing. And as you probably know, ancient DNA is usually sequenced using pretty short, let's say 75 nucleotide paired reads, relatively short reads. And there's a lot of damage in ancient DNA. Overwhelmingly, this is deamination damage, so C to T or G to A mutations. And overwhelmingly, they occur at ends of reads. Here, I'm showing a typical plot from our earlier paper published in 2019, where we plot frequency of these C to T or G to A mutations with respect to the human genome reference sequence at read ends from the left side and from the right side. And we see that there are very sharp spikes in frequency of deamination at read ends. And that type of DNA damage is actually more of a blessing than of a curse, because it helps us to bin reads into contaminant reads and genuine ancient reads, because nearly all genuine reads have deamination damage at their ends, while reads coming from modern molecules have very little damage. And the damage doesn't show these very characteristic spikes at read ends. That's why this type of damage is nowadays used very extensively for removing of contamination, which is very common in archaeogenetic samples, because most samples are excavated in the field with archaeologists who use their bare hands, then handled by anthropologists in collections who also use their bare hands, and so on and so on. Okay. But ancient DNA damage that is unrecognized or improperly removed, of course, may be a big problem for ancient DNA research. But I should say that I'm not an expert at all on this first stage of archaeogenetic research on DNA isolation sequencing, variant calling. I know the general principles, but I may not be aware of the latest practices in this field. And I have little personal experience here. Then, of course, there exist variant calling problems, such as reference bias, which is commonly known. Then batch effects. These are actually particularly important in archaeogenetic research, because archaeogenetic data sets come from multiple teams, and there's little standardization nowadays in the field. Some labs use predominantly shotgun, other labs use SNP capture, so enrichment of samples for certain parts of the human genome where interesting variable loci are located. And most commonly used panels of this sort are called 1240K and Twist. Both come from the Reich lab, but nowadays Twist is used, as far as I know, in other groups too, because it's commercially available, unlike this previous 1240K panel. But anyway, like maybe 2/3 or even more of existing archaeogenetic data from all over the world was generated on these two panels. And 1240K stands for 1,240,000 loci in the human genome, mostly on autosomes. And here I show a picture from this paper by Reich's lab, published in 2022, where they show distribution of loci, of SNP loci by coverage. And various panels have very different shapes of these distribution. The 1240K panel has this exponentially decaying curve, which shows that only a very small fraction of SNPs actually are sufficiently enriched there. So for getting good coverage on, let's say, 500,000 sites using this panel, we should sequence very deeply, actually. But Twist in blue has a profile which is much closer to that of shotgun genomes. And these differences in coverage distribution across sites, they actually have a major effect on various downstream analyses such as PCA or F statistics. That's why it's tricky to combine, let's say, shotgun data and SNP capture data, and moreover SNP capture data generated using different technologies such as 1240K or Twist, or even using the same 1240K technology but from different labs. We have this sort of experience. Okay, then let's say we do have variant calls of relatively good quality with a relatively even coverage across the genome. And now we can use those either for studies of population history or for functional studies of variants, let's say for statistical genetics, which is not my area of expertise. So I will talk today mostly about population history. And there's another type of bias, which is important for studies of population history, and that is so-called ascertainment bias, associated with a set of variable loci itself, even if they are perfectly sequenced. So while these batch effects arise due to, let's say, imperfect sequencing, imperfect coverage distribution across loci, ascertainment bias arises even under perfect coverage on simulated data. And here I'm showing some results from our paper published last year. Let's talk about F4 statistics. F4 statistics is a very popular tool invented again in Reich's lab in 2009 and published more properly in 2012. Unfortunately, I don't have a diagram here showing a typical F4 statistic, but imagine an unrooted tree of 4 groups. There are 3 possible trees of this sort with different arrangement of groups. The tree is unrooted again, and F4 statistics just measure correlation of allele frequency differences in these 2 clades on the tree. Let's say we have here African X, archaic individual, African Y, and Middle Eastern individual, a group. And these 2 form clade 1, these 2 form clade 2. And a F4 statistic for a single loci is basically A minus B multiplied by C minus D, where A, B, C, D are allele frequencies at this locus in these 4 groups. And under the assumption of cladality, under the assumption that this particular tree is true, this F4 is expected to be 0. And differences in F4 from 0, they suggest that there is a gene flow between at least 1 pair of groups in these 2 clades. Or if the deviation is very large from 0, it means that the tree is entirely wrong and that another tree is right. But basically, there's a continuum between gene flow and an absolutely wrong tree. Okay, but again, this is within the paradigm of trees. And later I will tell about another paradigm of isolation by distance. So here I'm just showing examples of 4 classes of F4 statistics on real data on 2 sets of sites. On the x-axis, on all sites from shotgun data, And on the y-axis, either on 1240K sites without missing data in this particular set of individuals, or on AT or GC mutations that pass a certain minimum allele frequency threshold in Africans. But that's not so important here to now pay attention to these details. So what is important here is to see how different are values of statistics in these four classes on all sites, and let's say on 1240K sites. And there are four classes of statistics. The first class, African, one, archaic individuals such as Neanderthal or Denisovan, African, two, and a non-African individual. And other types of statistics, for example, this one includes only one African and three Mediterranean or Middle Eastern individuals. And we see that for this latter class of statistics, all of their values lie actually within two standard error intervals from the diagonal. But here, for this type, nearly all lie outside of two standard error intervals. So this class is actually the worst class we found. It's extremely biased for some reason. There are also other intermediate levels of bias, for example, for this class, including 3 Africans and 1 archaic individual. Bias is very noticeable, but of course it's not as bad as here where all statistics are biased. Then we can move one level further from F statistics, and instead of these scatter plots, look at residual standard errors. So look at basically the magnitude of deviation from the diagonal in these scatter plots and plot it on the y-axis, and then compare various types of ascertainment. So on the previous plot, we compared non-ascertained data, shotgun data, and only one type of ascertainment, which is the 1240K panel, the most popular panel in RQ genetics at that time. But here we also compare other types of ascertainment to randomly chosen sites. So here on the left, we have two sets of randomly chosen sites equal in their size to 1240K on this set of individuals. And to the right of it, we have various other ascertainment types. And we see that for this particular class, all ascertainment types are biased. There's another class for which some ascertainment types, for example, this ascertainment on minor allele frequency in Africans is not biased. But this class, nearly all ascertainments, including 1240K, are unbiased. That is, the residual standard error lies below the 97.5 percentile of residual standard error on these randomly sampled sets of sites. And finally, we can repeat this analysis not for F statistics, but for admixture graphs, which are simple tree-like models with admixture that rely on F statistics. So instead of residual standard errors of these scatter plots, we plot rest residuals of admixture graphs. And we also see that for a graph that relies on this worst performing class of F4 statistics, indeed, all ascertainments are very biased. And here we can compare rest residuals of these admixture graph models on all sites and rest residuals on 1240K, for example. And we see that there are many outliers and the correlation is very bad. So each dot here is an is a particular simple historical tree-like model fitted to real data. But in the previous paper, we looked at the data from the perspective of admixture graphs. And admixture graphs are very common in the literature, a very common way of thinking about population history. And it relies on the idea that panmictic, relatively isolated populations are pretty common, and that simple models composed of these panmictic isolated populations with few admixture events on top of them, that these models are reasonable approximations of history. Of course, everybody knows that this is just an approximation, but people tend to think that it's useful and reasonably accurate. And there's another way of thinking about history in terms of isolation by distance landscapes, or in terms of stepping stone landscapes, which are approximations of isolation by distance landscapes. For example, I'm showing pictures from this paper introducing a certain method working on isolation by distance landscapes, and illustrated using genetic data for elephants. And there's a lattice, there's a triangular lattice overlaid over Africa, and demes or individuals are arranged on this lattice. So we have coordinates of demes or individuals on the lattice, and we can measure various properties of this structure. We can infer genetic barriers, like is shown here on the right. Genetic barriers are shown in brown, and areas with a pervasive gene flow are shown in blue. So there are these two different ways of thinking about history, and for reasons which are probably more due to history of science, they are these ways of thinking are rather compartmentalized across labs. So one labs almost exclusively work in the paradigm on the left. Other labs work almost exclusively in the paradigm on the right. And let's move on to other potential sources of bias, so far staying in the mixture graph paradigm. And these are modal identifiability problems. So even when the type of population history, such as a mixture graph, is well specified, when comparing alternative graph-like models of history, we run into multiple problems. For example, here we explore on simulated data in this paper, and on real data too, if we can uniquely find a best informative model of history with admixture graph fitting. So we try to run a search of the space of all possible admixture graphs using a certain model fit metric, and also comparing these model fits using Bootstrap. And in this central panel, panel B, we show proportion of newly found admixture graphs that fit significantly better than the true one. Four subtypes of models, 4 models with 4 or 5 admixture events, 8 or 9 leaves, so 8 or 9 linear, 8 or 9 groups on this tree, and 4 to 5 admixture events. And also with different number of events allowed while doing this search. So we simulated 4 or 5 events, but we can allow n-1, n or n+1 events. And we see that in a large fraction of these experiments, let's say 25 or more than 50% of newly found models fit significantly better than the simulated one. And as we illustrate in panel E, these are not simply small variants of the simulated one. These are actually large topologically diverse classes of models that have nothing in common with each other and nothing in common with the simulated model, which simply says that this particular approach, which became relatively popular in reconstructions of human history, and not only human, even dog, wolf, or horse history. This method is actually completely unsuitable because there's very poor modal identifiability even on simulated data, which ideally conforms to admixture graphs of relatively small complexity. And here's another illustration of this thing on real data. So we here show model fit scores, log likelihood scores for newly found models on various published data. So in all of these papers, there was at least one admixture graph model which was featured in the main text and which was used to support some of the main findings in that study. So it was not used as just an illustration of a finding that is supported by other methods, but it was actually used for learning something new about population history. And as red crosses, we show fit scores of best models we found. And as blue crosses, we show fit scores of published models on this data, on the published data. And with these dots and whiskers, we show bootstrap scores for the same models. And for example, in this case, we see that we found dozens of alternative models which fit nominally better than the published one here in blue. And the best alternative model, and actually this large set of alternative models, they actually fit significantly better than the published one. The green line shows the approximate boundary between models that fit significantly better or those that don't fit significantly better. And there's another example from another study by our team where we compare published model, a particular published model that was used to support some findings in the paper on population history of Southeast Asians, of human populations in Southeast Asia. The models here are plotted in coordinates of 2 fit metrics, log likelihood score and registry residual. And so the published model is in red, and all alternative models we found are in green. And these models with magenta borders, they fit significantly better than the published one, and the topology is very different from the published one and does not support the findings in that paper. So that's an example of this result that it's very dangerous to have any findings that are based exclusively on admixture graph models, because they're basically unconstrained by F statistics on which they rely. And finally, let's make a bridge to the second part of the talk on PCA. And this is an illustration of another problem in this paper by Bate and colleagues, where they compare a performance of various methods for visualization of population structure on high quality synchronous data. So on data collected at the same time, in this case on present day data for mosquitoes or for humans. Here I'm showing results for humans, and they compare PCA with a machine learning technique with so-called variational autoencoders on the right. And we see that PCA on this worldwide set of modern humans, which is actually the HGDP panel, a set of almost 1,000 high coverage modern individuals of various ethnic groups. On this set of individuals, if we just throw all individuals in, PCA looks like this, with Africans in gray, and so on. So individuals are colored by continents. And they also show biplots for other PCs here. And of course, this arrangement doesn't resemble the geography of our planet. It resembles it only in a very vague way. But this arrangement on the right is actually much more similar and geographically recognizable, much more similar to the real geography. Africans are here, then we have Middle Eastern individuals and European individuals above them, Central Asians and South Asians here, Americans here, which is actually perfectly explainable if we know a little bit about the settlement of America, East and Southeast Asians here, and Oceanians here. So this is actually almost a perfect map. And here the authors just compare geographical coordinates with coordinates of individuals in so-called latent space of this machine learning model. And here they measure correlation between geographic distance on a sphere and latent space distance on this two-dimensional latent space. Compare for 4 methods: PCA, variational autoencoders, t-SNE, and UMAP, which are also machine learning techniques as far as I know. Compare the correlation of geographic and latent space distances under settings that are the best for revealing global structure, because it's widely known that t-SNE and UMAP are very good at revealing local structure, but pretty bad at revealing global structure. So here it looks like the R-squared is almost the same for t-SNE and this variational autoencoder, but it's actually just an artifact because the authors measured just distances on a sphere, ignoring oceans and other features of the landscape. But for example, here is an embedding produced by t-SNE under these best settings. And we see that Americans here lie on top of Africans. So the sequence is Middle East, Europe, Africa, and America, which is absolutely wrong. So in this case, there is some correlation with geography on the x-axis, but actually no correlation with geography on the y-axis. And maybe there's local correlation with geography, but there's no global correlation with geography. That's why, at least in this paper, it was clearly shown that variational autoencoders outperform BCA, t-SNE, and UMAP in revealing correlation of genetic and geographic distances. So that's the end of the first part of my talk. Do you have any questions?
Okay, I think that, well, if you don't mind, we can spend a little time with discussion, because I'd like to jump in with a bunch of questions that, yeah, I think that they're relevant to this, and then we'll proceed to the second part, which is more, as far as I understand, the unpublished part. Is that okay?
Yeah, yeah.
Okay, good. Yeah, so maybe then I can start by returning to the maybe the early parts of what you've shown. So we've basically shown that there is ascertainment bias and many differences relating to the actual way out the data were compared. So maybe so basically, the question which I think is very important in this sense, which bothers me, for example, is whether or not it's actually a good idea to do some comparisons, with data that have been obtained using different methods, and like the best practices which can be done in order to ensure that you have relatively harmonized data, so that the source and method that has been used to get the data actually does not have a tremendous effect on the results you get. And I think that there are two parts to this thing. So first there is the issue of just again, data obtained from various sources or from various methods, like SNP capture or genome-wide and others. And another thing is integrating the contemporary genetic data with ancient data. So if you could maybe, as a first part of the discussion, maybe elaborate a little bit on what you think is a good way to ensure, again, that the data that you work with are harmonized, and whether you all consider it a good idea to do these sorts of like integral and integrated kind of analysis when you take data from obtained with different technologies and test and hypothesis using these kinds of heterogeneous data.
Okay. So for like, I don't think I'm comfortable to give general advice here, because this topic is very broad and quite a lot has been done in this small subfield. So I can definitely say that it's okay to take a big data set generated by a big lab. For example, this well-known Allen Ancient DNA resource from the Reich lab, which is basically a compendium of published archaeogenetic data generated using all kinds of technologies, but restricted to the 1240 case sites. So again, we should always keep in mind this difference between site set and technology, because both can cause bias. Technology can cause bias, as we said, due to mostly this effect, while ascertainment bias, in the case of F statistics and other analysis relevant for population history, it affects results. But it affects results, as we showed in this publication, almost exclusively when we analyze Africans. So when we analyze many Africans together, especially Africans with archaics and or non-Africans, then we run into problems. But when we construct BCA or calculate F statistics or fitted mixture graph models that are based on F statistics for non-Africans, for all kinds of combinations of non-Africans, there's no bias. So at least if you construct BCA or run F statistics for non-Africans, or let's say one African outgroup and all the other groups are non-Africans, there's no problem. You can use basically any kind of SNP set. Of course, I've not tested some extreme cases, but cases that are popular in the literature are all reliable in when we restrict our analysis to non-Africans. But when we restrict analysis to Africans for reasons which I will not go into here, the results are much more variable, and they depend a lot on a particular set of SNPs. And this is visible even on PCA, because if you, let's say, take a certain worldwide set of individuals and then start pruning sites in linkage disequilibrium in various ways, and then run PCA for each way, and the only part of the world that is variable is actually Africa. The rest of the world is stable. And I think that's a manifestation of the same thing, because PCA spaces and F statistic spaces, as was actually mathematically shown recently, are linked. So many things that we see in PCA spaces, we see in F statistic spaces too. But it's hard for me to recommend how to co-analyze, let's say, ancient and modern data for, let's say, analyzing copy number variants, or for running GWAS and so on.
Yeah, well, actually, with regard to structural variation and other types of variation, I think this is quite tricky to do this sort of aggregation with archaeogenomic data, especially. Yeah. So I'm not maybe talking about these kinds of variation, but still, yeah. So as far as I understand your answer, well, the idea is that. First we can the concordance shall be, as far as I understand, better for the variance in the common panels, for these sets of sites, and secondly, you say that there is this issue with the African individuals. So again, for those in the audience who are maybe less familiar with the specifics of human genetics, the idea of the African population is that it's actually much more diverse genetically compared to other populations. Correct me if I'm wrong.
Sure.
The question, which I think maybe is a follow-up here, will be when you see this effect, is it actually due to the fact that there is more diversity within Africa, or what is the problem here? So maybe as an outlook to studies which involve non-human organisms. So I know there are some people, for example, in the audience who work with the non-human data. So if you maybe say a few words about what is actually, do you think, wrong with the African data in this context? So that what shall be done, in order to reduce bias and maybe other types of other kinds of non-human data in particular.
Okay. So I did want to show these figures from the paper because they're pretty complex, but I can explain here. I can try to explain here in a few words. So the point here is that it's important to capture the genetic diversity that existed at the root of all populations being co-analyzed. For F statistics to work in an unbiased way. And let's say the 1240K panel shown in green here. This panel is a non-principled construct, actually, although it may sound in some papers that it was constructed in a very principled way. This is absolutely not true, because approximately half of the sites come from the human origins panel, another older panel from the Reich lab that was used exclusively for genotyping modern individuals for the purpose of population history studies, so usually not for medical purposes, but for exclusively historical purposes. And that panel was constructed in a way that was at least somehow principled. So sites there were ascertained as heterozygous in a single high coverage genome from one population, and then several populations, let's say 13 populations from all over the world, were combined. So first, sites were ascertained in, let's say, one Sun individual, one Yoruba individual, one British individual, and so on. And then all these sets of sites were combined. That's human origins, and we tested this type of ascertainment on simulated data too. But then approximately another half of the sites in 1240K come from a particular Illumina panel. And an Illumina panel is a very complex construct with some medically relevant sites, with some historically relevant sites, but mostly based on variation in Eurasia. So not in Africa. And as an end result, more than half of sites on 1240K are almost invariant in Africa. So they don't represent African variation well. So the allele frequency spectrum that existed at the root of all the groups being co-analyzed is very skewed on 1240K. So so it's better to ascertain as close to the root as possible. But on the other hand, ascertaining very close to the root, let's say we have access to a very ancient population that was close to the root. We ascertain on it. And so we choose, by ascertaining on it, I mean we choose sites that are polymorphic in it. And then we genotype these sites in all the other groups and do some analysis. In that case, the analysis will most likely be unbiased, but the sensitivity will be pretty low. Because again, we lack some variance that arose after the root. And we cannot differentiate some groups very clearly because we have access only to sites that were polymorphic at the root. But if you read Nick Patterson's 2012 paper on F statistics very carefully, there is a note there that F4 statistics and other F statistics are unbiased if polymorphism is ascertained at the root. But people, when doing further work, actually, people ignored this note. Even Nick Patterson himself ignored this note, but actually it's important.
Okay.
But the take-home message here is that if we actually ascertain on Africans, so if we take a diverse collection of Africans and just take variants that are common there, so we remove rare variants, we take just common variants, let's say with minor allele frequency about 5%, that's actually the best type of ascertainment, which is shown with red border here. So it's unbiased for nearly all classes of F4 statistics, except for this strange class for which everything is biased. But it's unbiased for all the other classes.
Okay, I see. Yeah. So maybe as a follow up for this, the F statistics and mixture graphs. I may be also referring to some results which have been shown a little bit later during your talk. So when you've shown that, for example, you can have for simulated data, the models that fit better than the actual model that was the data was simulated. So here I would like to maybe, and that may be a segue into your second part of the talk. If so, that we can proceed. If not, then you're free to elaborate before that. So the question is, like which way of doing the actual history analysis, the reconstruction of history, is best in terms of which methods are the most informative, or which combinations of methods should be used. For example, the F statistics, naturally, the PCA, which I think you will also talk more about, and there are other methods. So, for example, one thing which I really wanted maybe to hear your opinion on is the genetic clustering algorithms, such as admixture, for example, which do the analysis of trying to basically just deconvolute each individual with like proportional contributions from different ancestral populations. So maybe you can say a few words, and again, if you feel that it would be better to do so after you finish the second part of the talk, then we can move on. What's based actually, given all of the things that you've already shown, the best maybe pipeline in terms of actually picking the methods and the maybe priority of in which you should apply them in order to get a good picture.
Okay, so so I think it would be better indeed to talk about that at the end of my talk.
Okay.
So these results are yet unpublished, but I've been working quite a lot on this in recent years, and like the main take-home message here is that what matters is like first, hypothesis testing makes no sense when we reconstruct population history. Okay, it makes little sense because people often try to let's say compare models. They arrange models of various topology with various branching order, with various admixture events. They arrange these models according to certain criteria of model fits, such as worst residual or log likelihood or anything else. And they try to rank them, try to pick the best model, the best class of models. Sometimes they even run bootstrap comparisons of statistical significance and make some conclusions. They reject certain models of history or accept other models of history. But there's a very deep problem that we don't know, or if the basic assumptions about history are correct. If the assumption that admixture graphs are informative and not misleading, or more informative than misleading, is correct. And all my work shows that this is actually wrong. The graphs are more misleading than informative. And especially they are misleading even if the underlying history is graph-like, as I showed in this paper. But if the underlying history is non-graph-like, so if it more conforms to isolation by distance landscapes, which I will talk about in the second half of the presentation, then applying admixture graphs is just a useless exercise. And the same is actually true for not only for graphs which are like a high-level analysis based on F statistics, or based on site frequency spectra or other type of data. This is true for F statistics themselves. This is true for admixture. This is true for QPADM. This is true for PCA. This is true basically for all methods. So either they don't make any sense like graphs in the isolation by distance for framework, or they should be interpreted with this framework in mind. Otherwise, the interpretation may be wrong. But this is a contentious area of research. And as I said, some groups in the field actually for many years have been working in the isolation by distance paradigm, while other groups such as the Reich Lab and many other well-known archaeogenetic labs, they work in the admixture graph paradigm. And they don't work together too much, I mean, trying to merge these paradigms. For example, nowadays, there are even these studies on horses or wolves or dogs, like ancient horses, ancient wolves, and other animals that just take this standard archaeogenetic toolkit that is PCAF statistics, QP, ADM, admixture graphs, in the admixture graph paradigm, take that toolkit and just, without any changes, just transport it to another species, to horses, and apply it there with absolutely wrong conclusions. So the conclusions are just completely wrong, because horses are even more mobile than humans. That's why genetic variation of horses cannot be modeled as a tree or a graph. It's just a smooth landscape. Of course, broken sometimes by environmental barriers, especially if we consider long history.
Okay, I suggest that we then move on to the second part of your talk, and then after that I will maybe a little follow up with some more questions on this thing. Yeah, thank you.
Yeah, I don't think we have so much time, so I will try to skip some slides. So, okay, so I decided to talk about PCA because PCA is a method we all use, and for getting an overview of population structure, and it's widely used in both historical and statistical genetics. In statistical genetics, it's used for stratifying groups, let's say for stratifying cases and controls by their ancestry profiles and so on. And this method is not a black box, unlike, for example, these variational autoencoders. Unlike machine learning, it's not a black box. And there are papers like these two, which link PCA status to coalescent time and app statistics. For example, in this paper, it was shown that there exist relationships between app statistics and PC spaces and arrangement of individuals in PC spaces. But is PCA an optimal method for visualizing population structure? And that's what I'm interested in. So as highlighted in the paper on variational autoencoders, PCA reduces dimensionality to S-1 or V-1, whatever is smaller. And S is the number of samples, and V is the number of variants or sites or side blocks. But this dimensionality is still very high. And the signal thus is spread across many dimensions, and in nearly all papers, just the first two are interpreted and shown. But what is natural or true dimensionality of genetic variation data? That's probably a bad term, but let me use it. So, and I contend that it's just 3. If we are talking about neutral variation at the population level, and in the absence of batch effects, so in the absence of various sequencing technologies and missing data, because individuals exist and mate in 2D space and 1D time, in 2-dimensional space and 1-dimensional time. So if you want to reproduce a certain history of a species that with free interspecies mating, Ideally, we should do it in 3 dimensions. But is this a problem for PCA? And also, is poor sampling a problem for accurate visualization of population structure with PCA? And to explore that, I introduced these simulations, which I term stepping stone landscapes. And these are approximations for isolation by distance landscapes. And this landscape, stepping stone landscape, is a genetic history consisting of at least one colonization epoch, approximated as a tree, or more generally a directed acyclic graph, followed by at least one isolation by distance epoch, which can be approximated as an undirected graph on the right, where each edge is a bidirectional gene flow edge. And in our simulations, we usually sample intensities of these gene flows from certain uniform or normal distributions, so that the intensities of forward and reverse flows are allowed to vary in space and time. And but first, let's look at PCA patterns on random trees composed of 40 genes. So we have 40 branches sampled at one point in time at the end of the simulation, here on the left, or sampled at various points in time, here on the right. And we can see that they all conform to basically the same pattern. These are rays of individuals fitted into a triangle in 2 dimensions, tetrahedron in 3 dimensions, and so on. That's a very common pattern we see in PCAs on real data too. But how should we interpret these patterns on real data? Let's now look at PCAs on directed acyclic graphs, so histories that are shaped as directed acyclic graphs, again with 40 branches sampled and 15 admixture events in that history. We show results for 10 random graphs on the left and 10 random graphs on the right. Again, sampled either at present on the left or throughout, or at various time points on the right. And again, we see that the pattern is usually more complex, but again, all these shapes in two dimensions are embedded in triangles. Now let's move on to very simple isolation by distance or stepping stone landscapes, namely 2 one-dimensional landscapes, for example, explored in this important paper published a long time ago in 2008 by November and Stevens. And they show that if we take a one-dimensional string of DEAMs with gene flows between nearest neighbors of the same intensity between nearest neighbors, and then construct PCA biplots, let's say PC1, PC2, or PC2, PC3, PC3, PC4, for this set of individuals, they all lie on so-called Liajoux curves. And if we also add undersampling or oversampling, for example, if we oversample the center of this simulation and undersample the left arm of the simulation, we get a bit distorted shapes. But for example, this shape on the left looks very similar to worldwide PCA, to some types of worldwide PCAs for humans. And we just repeated this very simple analysis. On the left, we show PCA for one dimensional landscapes, but sampled at 3 different points in time, from old to young. And again, we see just a series of concentric horseshoes. So this PCA pattern is, of course, distorted with respect to the original landscape, but it's easily interpretable if we know what a horseshoe effect is. The horseshoe effect is very common. It appears whenever smooth gradients of any sort exist in the data. It's not unique to genetic data. It appears in voting data for US Congress and for many other types of data. Whenever smooth gradients exist in the data, it appears. And whenever the data is approximately one-dimensional, so not strictly one-dimensional, but approximately one-dimensional. But interesting things start happening when we increase the sparsity of the landscape. And by sparsity, I mean the intensity of gene flows between the points that we sample on the landscape. So we can either keep the size of the landscape the same, but decrease the range of gene flow intensities that we simulate on that landscape, or we simulate a large landscape with intense gene flows, but then subsample it more aggressively. And here on the right, I'm showing PCAs for a very sparse landscape, which is one-dimensional. And these PCAs look more interesting. So here, horseshoes are not always recognizable. And again, I'm combining individuals from various sampling points in time. They're shown with different marker shapes. But the same effects we see if we take only one slice in time. And this type of system is more illuminating to explore in 3 dimensions, because if we look at 3-dimensional PCA on this sparse 1-dimensional landscape, it's easily visible that actually there's one gradient that is just twisted and curved. But if we here now look at the PC1, PC2 projection, it's less clear what's going on. While, again, if we rotate in 3D, it's clearer what's actually going on. So sometimes it's more illuminating to examine PCAs in 3D, which we now routinely do for real data. But it doesn't solve all the problems, of course. Okay. Now let's move for some time away from PCA, and let's combine isolation by distance and identity by descent. So 2 IBDs. What is identity by descent? Identity by descent, that's basically a name for long autosomal haplotypes, which can be relatively easily called on modern high coverage data. And that type of analysis allows to look at population structure in a very fine-grained way. For example, there was a study in 2015 looking at regional variation in Britain, which they were able to do in a very, very detailed way. And only recently, this method was introduced to archaeogenetics with the recent advances in imputation. So now there's a flurry of papers coming out, including our own manuscripts in the pipeline that use these long autosomal haplotypes or IBD segments for various kinds of analysis on ancient DNA. And again, I'm talking about human ancient DNA. But I'm pretty sure that soon this method will be used for other species too. And we use a method which is very simple, and we term it IBD graph. And here we compare reconstructions of populations to rupture using IBD graphs and using various kinds of PCA. So we simulated a very simple, nearly one-dimensional landscape composed of 22 DIMs arranged on this triangular lattice. And again, with bidirectional gene flows between them and with randomized gene flow intensities. And then we sampled each DIM relatively densely at 5 points from 0 to approximately 2,000 years before the end of the simulation, so before present in simulation time. And on the left, I'm showing reconstructions of networks of IBD connections between these individuals using two approaches, either by when we remove links between individuals that are too distant in time, or we keep all links. And we also run PCA for the same sets of individuals without projection on ancients, on most ancient or modern individuals, or with projection on present-day individuals, with projection on ancient individuals. And projected individuals are here separated from non-projected individuals to make the figure clearer. And then we repeat this analysis for an approximately 10 times higher gene flow intensity on this landscape, and we see the patterns are actually different. And I want to look at the most important thing here. So first, we see that PCA without projection, of course, it resolves the longer spatial dimension of this landscape. So this landscape has two dimensions, the longer one and the shorter one. The longer one on X, the shorter one on Y. And of course, PCA at various levels of subsampling can easily resolve the longer dimension, which is coded here by this color gradient. But it has troubles resolving the shorter dimension, which is not surprising. These DIMs exchange migrants pretty actively, and they're pretty similar to each other. But if we construct network graphs where nodes stand for individuals and edges stand for IBD segments shared by these pairs of individuals, then we can sometimes achieve much better resolution of populations to rupture. For example, here, when we don't filter the IBD segments in any way except for taking long IBD segments longer than 8 centimorgans, we can see that we resolve clusters corresponding to each simulated DIM. These clumps are actually correspond to simulated DIMs. And the point size here stands for the date of the individual. So we see that old individuals, large circles, and young individuals, small circles, they are intermingled here. Or if you look more carefully, we see that this dimension of time is actually curled up in this layout. It's curled up in each cluster corresponding to each DIM, but the dimensions of space are accurately visualized. If you want to visualize only the predominant dimension of space and time, we can remove individuals we can remove IBD links between individuals whose dates are very different, let's say more different than 650 years on human time scale. And immediately we have the longer spatial dimension on X and temporal dimension on Y. So we can follow the evolution of the system on the Y-axis and the geography of the system on the X-axis, but we lose the smaller spatial dimension. And but if we have higher gene flow intensity, here there's basically no smaller spatial dimension, and that's why filtering on day difference has very little effect. But when we look at PCA, we see that, of course, PCA, it reproduces the same geography and the same temporal gradient too, but they are more distorted, especially if we take fewer individuals. But the same is true for these IBD graphs, and actually IBD graphs fall apart faster under sparse sampling as compared to PCA. So for accurate reconstruction of isolation by distance landscapes, we need very dense sampling in space and time, but we can approach this problem successfully with IBD graphs. But here and in later parts of my talk, I contend that we cannot approach this problem with PCA. For another problem of PCA, besides these widespread horseshoes, is emergence of outliers. For example, they emerge here in non-projected ancient individuals, or they emerge here in when all individuals are co-analyzed, both ancients and moderns, and they emerge also in many other cases. And these are artifactual outliers. I will also talk about them later. And now let's move back to previous one-dimensional simulations. But first, let's talk about the nature of archaeogenetic sampling. Unfortunately, the kind of sampling we are dealing with is sparse and nearly random, because a substantial fraction of ancient human or animal populations remains archaeologically and thus archaeogenetically invisible due to various reasons. First, there may be burial rites that leave no suitable remains, such as cremation or burial on or above ground, and poor DNA preservation conditions such as acidic soil or hot climate also skew our sampling a lot. And biases in archaeological research are actually very common. This research is often driven by salvage excavations in industrial or agricultural landscapes, but in other landscapes, almost nobody digs, and or even by political agendas. And even when we talk about existing anthropological collections, we often lack systematic sampling of those. Thus, for the purpose of this analysis, I contend that our existing archaeogenetic sampling of humans and I think most animals is nearly random and very sparse with respect to the effective sizes of the populations that have ever lived. Okay, now let's explore the performance of PCA under subsampling. Let's say we simulated again this simplest one-dimensional case with 40 demes and with effective size of 1,000 diploid individuals for each deme. Then we sampled 5 individuals per deme per sampling epoch. Here we sampled from 3 epochs, so we have 600 individuals in total, and then we take only 60 of those randomly selected across epochs and co-analyze. on PCA, again, without any missing data, without any other confounders. And we see that this beautiful horseshoe pattern collapses into triangles and rays embedded into triangles. And we've already seen this pattern actually before, and this pattern we saw when we simulated admixture graphs and trees. So in PC space, under aggressive subsampling, the data becomes indistinguishable on landscapes and trees. So we lose information about the kind of process that generated the data. Now let's move on to more complex landscapes. Here we simulated an approximately circular landscape composed of 64 demes and originally rising via multiplication, but then evolving for approximately 2,500 generations in this stepping stone mode, but with varying gene flow intensity in space and time. And then we explore 3 types of landscapes with high gene flow intensity per gene flow per generation on the left, with intermediate in the middle, and with very low 10 to minus 5, 10 to minus 4 per generation on the right. These numbers mean that 10 to minus 5 to 10 to minus 4 of individuals in a particular deme in a particular generation come as migrants from a neighboring deme. And here, first we explore PCA without any subsampling, just to show these patterns. And we immediately see that, of course, there is a stratification of old and young individuals, of individual sampling at various time points. And in the high gene flow intensity scenario, there's actually very little in common between these 2 time points. So they form separate clusters often in this PC space because they're very, very different. Because here, the time points were several hundred generations apart. But what happens if we subsample these PCAs? Of course, all these analyses are not exhaustively sampled with respect to the cumulative effective size of this simulation. But here we sampled 5 individuals per each deme at each time point. And here we take just 1/10 of those individuals randomly. And we see that under the high gene flow intensity scenario, of course, we don't get any more this perfect circular pattern, but sometimes the original landscape is recognizable. But as we move on to sparser and sparser landscapes with lower and lower gene flow intensity, given the same landscape size in demes, we move more and more into this tree-like territory where PCA patterns look like rays embedded in triangles. And this is the type of pattern that we get from trees or grass. And the same we illustrate here, but without this confounder of 2 epochs of sampling. If you take only one sampling epoch, we see the same effect. In the case of the sparsest landscape, the PCA is no longer representative of the original landscape. While in the case of the densest landscape, it's more or less representative of the original landscape. And again, we saw this pattern before, and this triangular ray-like pattern is typical for PCAs on trees, on random trees or random and mixture graphs. Now, let's look at another type of PCA that is now basically standard in archaeogenetics. It's a gold standard in archaeogenetics because previous analyses, they were based on co-analysis of old individuals, so ancient, modern individuals, but usually, like in this very well-known paper by Haag, 2015, ancient individuals are projected using LSQ projection. So data for ancient individuals are projected on axes on the PC space based on modern genetic variation. That is done allegedly to get rid of problems associated with high levels of missing data, and also partially to get rid of problems associated with batch effects. And here, I will skip this. So we explored 3 kinds of projection algorithms, because in Nick Patterson's Smart PCA, there is so-called shrinkage correction algorithm that is almost always used for adjusting the positions of non-projected individuals in such a way that projected individuals don't look like a small blob in the center of the original space. For example, here in this pair of panels, we show a projection of all ancient individuals on a random subset of 50 modern individuals. So PCA for this random subset is here. An identical PCA we get when we apply Smart PCA with the auto shrinkage correction algorithm. But ancient individuals all fall into the middle of the space. And of course, that is not very attractive to have this property if you want to co-analyze ancients and moderns in any meaningful way. So for that reason, this new shrink algorithm was developed, which naively one may think that it changes the positions of projected individuals, but that is not true. It keeps the coordinates of projected ones. If you compare them, they're actually the same between this panel and this panel, but it changes the coordinates of non-projected individuals. But as we show here on this random small subsample from this actually very big landscape, composed of more than 300 demes, we see that it completely reshuffles the set of non-projected individuals, while the auto-shrink algorithm, which was introduced before new shrink, so it's less advanced than new shrink, it actually preserves the original PCA, while the new shrink algorithm preserves the projection, which is actually pretty much distorted, but moreover, it completely reshuffles the set of non-projected individuals. And the original coordinates on the landscape where the sampled individuals come from are shown here in this panel. And let's look at some other subsampling iterations. So we just, from the same simulation, we take just a little bit different set of individuals for constructing this basic PC space and then for projecting onto it. And here we see that a very common pattern is that the originally hexagonal landscape that is perfectly visible on a PCA without any subsampling, it becomes distorted into a hyperbolic triangle. And this is a very common pattern on simulated data under subsampling and with projection. And we see very similar things on real data. For example, here we project human individuals who lived between 3 and 1,000 years in Europe onto the same region, but in the Neolithic, so several thousand years before. And here we color them by geographical coordinates, by longitude and latitude. And we see that we see a similar shape where one side is hyperbolic-like, and similar triangular shapes we see in many, many other cases. For example, when we here we actually don't project when we co-analyze Europeans and Siberians and other Asians and Americans, or where we project various ancient Europeans and Siberians and Central Asians and East Asians onto modern variation in these regions. Again, we see something very similar to a hyperbolic triangle, and I'm pretty sure that this space is, so the effect we see here is identical to the effect shown here on simulated data, because the same projection algorithm was used, and we are dealing with a landscape that is sampled in a nearly random way and is sampled very sparsely if we are talking about ancient or modern individuals, because modern individuals are also very often not well representative of ancient variation. Okay, so I will skip further slides, and okay, actually I'm nearing the end of my talk. So, so is PCA suitable as a method for visualization of population structure? First, as we mentioned, dimensionality of embeddings is too high. And that's why PC1 versus PC2 plots are often severely distorted with respect to the underlying topology of the landscape. And machine learning techniques such as variational autoencoders can embed into an arbitrarily small number of dimensions, for example, 2 or 3, but they are known to reproduce global structure very poorly, apart from VAE possibly, as shown in that paper. And they are black boxes. That's why probably people don't like them too much, at least in archaeogenetics. But they are very popular in some other fields like single-cell RNA. And sometimes these methods also used in population genetics. Here I'm showing this famous figure from the All of Us paper released this year. And they decided to show Americans not on PCA, but on a UMAP plot, which actually caused a lot of controversy because this space has very little connection to ancestry, but it does show some local groups. And PCA, moreover, is known to be sensitive to even slight outliers. That's actually a well-known problem. And we see this very clearly when performing PCA on small sections of landscapes. Remember where I showed landscapes with high gene flow intensity, which is equivalent to taking just a small section of a bigger landscape with low intensity. And there we see usually many outliers just popping up. And also the same thing we see on small random subsamples. And the very sparse sampling of all individuals who ever lived, which is typical for ancient DNA research, 2D landscapes visualized with PCA collapse into triangles and ultimately into 3 rays in 2-dimensional PC1 versus PC2 space. And these patterns are indistinguishable from PCA patterns on random trees and graphs. And since PC space is connected to F-statistic space, it's no surprise that tree-like and graph-like models fitted to F-statistics usually fit archaeogenetic data very well. And these problems become even worse when LSQ projection on modern individuals is used, which is nowadays a standard in the field, especially when modern individuals underwent demographic collapse like those in the Americas. That's why in many papers on archaeogenetics of Americas, people don't show PCA at all, or show like very specific kinds of PCA, because it's a well-known fact that PCA doesn't work well for American humans, because if we project on moderns, that's a very poor representation of Americans because of genocide and disease and so on. So what solutions do we have? We could use IBD network graphs that I introduced, but they require dense sampling in space and time and high coverage data for imputation and IBD inference. TSNE and UMAP probably don't work, although I don't know of any exhaustive testing of their settings on landscapes. And these autoencoders probably work the best, but it's a black box. It's computationally very intensive. It requires a lot of GPU power, and the shape of the neural network and the starting seeds and training duration need to be optimized. And moreover, it was not tested on non-homogeneous, low-quality ancient DNA data. It was tested and shown to be very good only on high-quality data sampled at one time point at present. So as a conclusion, I'd like to say that sampling a non-negligible fraction of individuals who have ever lived is probably even more important for reconstructing history than achieving high and uniform sequencing coverage. And that is often ignored in the literature, and I previously shared the same opinion that it's okay to sample a few individuals here and there, and then construct various mixture graph models, or look at F statistics, or look at PCA, and somehow make conclusions about history. But actually, this is not true. For robust conclusions about history, if history conforms to a landscape, which is another topic, but I think that it does much more conform to a landscape than to a graph, then it all makes no sense. Then we need to have very dense sampling in space and time for reconstruction of these landscapes. And so this is true not only for PCA, but for many other popular methods. Thank you.
Yeah, thank you so much for the very interesting results. Yeah, yeah. If you let me, I maybe follow up with this idea of what to do with regard to we have data which are of course not sampled the way we want them to, or that they're not that dense in both space and time. Yeah, and if under these circumstances, yeah, if you earlier you mentioned that you have to bear in mind the isolation by distance landscape concept, and interpret the results in this framework. So maybe if you can summarize, if in a few minutes, what can be, again, given the fact that we understand that the sampling is not perfect and very far from perfect, which methods will be best, and like what kinds of analysis would you suggest as the sort of a good practice for history reconstruction? Again, given that we have uneven space sampling, uneven time sampling, so what would be the priority of different methods?
Okay, so I would say that currently, so as I said before, I would avoid any hypothesis testing formal statistics methods. They became very popular in the field. Let's say comparing admixture graph models based on site frequency spectra, or based on F statistics, or F statistics dam cells, which can be used as formal tests for admixture, or QP-ADM, which is another abstraction over F4 statistics, and also used as a tool for finding optimal admixture models, which we also explored in a large study, which is still unpublished, on landscapes. And all of these methods, they sound interesting, and people often say, okay, we use formal statistics to prove or disprove. But unfortunately, in these conditions, if we are dealing with landscapes, if we are dealing with poor sampling, this hypothesis testing is unreliable completely. So we often just overfit or test noise. So I would avoid any formal model comparison methods. I would avoid any formal statistics saying that, okay, this model of history fits significantly better than that one. Of course, there are circumstances when that is indeed the case. There are very clear-cut cases, for example, the migration of Neolithic farmers into Europe 8,000 years ago, or in some places of the world, migration of Yamna culture people or Kordilware people. These are like very clear migrations grew from sources that were previously isolated for at least some time. And in those cases, indeed, all these methods are very well applicable. And that's why they actually became so popular, because they were first applied to these very simple cases. And they produced results that are largely true in those cases. But not all of human history conforms to these things. And most of human history is actually different, as I think. I may be wrong. And but if we, let's say, talk about other species such as horses or wolves, I'm pretty sure that all of their history is different from isolated panmictic populations. So I would work with methods that treat individuals separately, such as BCA or IBD sharing graphs or autoencoders, or any other method that treats individuals separately, that doesn't group individuals. And I would work with methods that are aware of the dimensionality of the space. For example, I think IBD graphs is a very nice method that allows to reconstruct the topology of landscapes perfectly. But unfortunately, it requires very dense sampling in space and time and high quality data for imputation and IBD inference. And moreover, it also works very well on graph-shaped histories. I've not shown it here, but actually PCA for many graph-shaped histories, again, looks completely uninformative. Looks like a triangle, which is completely uninformative actually about the history, about the underlying history. But the history is just visualized. Like you see the graph that you simulated. You basically see it. It's a bit distorted, but you see it. Why is that the case? Because IBD sharing graphs, these are basically noisy pedigrees. So each edge in this graph is a link between distant relatives or close relatives, but mostly between distant relatives. So we can think of it as a very, very noisy pedigree. But if we then apply a force-directed layout algorithm to this graph, and also, as I said, filter it in a certain way, we can visualize both space and time. So unlike PCA or variational autoencoders or any other method, this method is aware of pedigrees in this noisy way. That's why I think it may be a very important method in the future, but so far it's not applicable to all parts of the world and not applicable to all species, and it requires high quality data. If we don't have access to these conditions, I recommend using PCA and admixture, which I have not talked about, which is probably my mistake, because I should have talked more about it, because it's also a very popular method. And what admixture does on landscapes, it basically cuts them into approximately equal sections. So if you have a landscape, then it cuts it into sections. And the same we see on real data as well. So it's possible to, I can actually, we show one slide from a different presentation. So there's actually a certain way to construct PCA for real data that is that reveals the landscape much better than a typical PCA does. And that is when we, from each, let's say if you're talking about human history, from each period, we take the oldest individuals not affected by Neolithic movements, which disturb the landscape a lot. So in that period, the landscape is in turmoil, and then another equilibrium is established later. So if we try to avoid this period of turmoil and look at the earliest individual from each region, and also we try to cut these regions in a smart way. Let's say we take river basins and so on, mountain ranges, and certain ecogeographic regions that make historical sense. And each of those regions, we try to downsample to the same number of individuals. Then we can actually construct a PCA like this, that where there's a very good correlation between geography and position in PC space, much, much better than on regular PCAs, and similar to that we see for variational auto encoders on modern data. And let's say then we take, what is this? Yeah, then we take admixture on a simulated landscape, and we show by color fraction of a certain ancestry component. And we see that it just cuts this simulated landscape into pieces. And the same we see on real data. Let's say we take the certain gradient we see in Eurasia and analyze it at k equals 2 or k equals 3, or we look at all of the world at k equals 4, or we look at all of the world at k equals 8, and we just see that if we interpret this as a landscape, then admixture just cuts this landscape to approximately equal sections. Is this informative? In some way, yes. In other ways, this is trivial. So, but this is unsupervised admixture. So, but in our experience, combining supervised admixture with IBD graphs with PCA constructed in this particular way can be actually very informative for placing individuals in appropriate ancestry contexts on a landscape. But it's hard to do, it requires a lot of work, and no hypothesis testing, no p-values here, unfortunately.
Yeah, I think that's a good take-home point about no p-values. Maybe also a question which I personally am interested in regarding the essentially the choice of sites for this kind of analysis. Yeah, because I think that in many cases, in many other kinds of analysis, people are very concerned with LZ, for example, and so that you have to prune the data set very significantly, and also do some choice regarding the frequencies of variance, etc. So if you can say a few words maybe in terms of the actual real world data set, Now, what kinds of filters in terms of frequency and LD would you apply in these kinds of analysis, or are there any, or you can just like plug in like all well genotype sites and that's it?
If you're talking about PCA, if you're talking about admixture, some kind of LD pruning is needed.
Mm-hmm.
I'm not actually sure like what level of LD pruning is needed because I've not looked at that myself in detail, just copied protocols from some other papers. But if you're talking about PCA, we ran some tests for this, and actually LD pruning is not very important. Again, basically identical patterns we get for non-African humans, whatever kind of LD pruning we use, whatever kind of software we use. If we use Blink or Smart PCA, if we prune aggressively, if we don't prune at all, the pattern is very stable for non-Africans. It becomes a little bit variable in the case of Africans. So this African tail, which we see here, it varies depending on the choice of sites. So the choice of sites can be affected by pruning by many things, by minor allele frequency filters and so on. But for non-Africans, these patterns extremely robust to the choice of sites. But if we do projection, let's say if we do projection with this new shrink algorithm on present-day individuals, you simply cannot avoid pruning because it won't work on very large sets of sites. It just doesn't work, or it's very, very slow. So for PCA, I don't think for PCA on non-African humans, it doesn't matter. On Africans, it matters a bit. Again, it's hard to translate these findings to other species, but you may think about it, how these findings are translated to other species.
Okay, yeah, thank you. I think that we can, because we are a little bit over the time, so we can conclude today's meeting. Thank you again very much for your time and for the talk, presenting very interesting results and sharing some of the well-valued experience. Thanks for everyone who is still with us. And see everyone next time. Павел, если можно еще секунду по поводу остального, я на этом моменте просто запись выключу. Да, я прямо сейчас ее выключу. Так, ладно, потом. По поводу просто того, что я скидывал, удалось посмотреть, нет?
Я посмотрел вот буквально